So, I'd like to introduce you to our keynote speaker. It is my pleasure to introduce Dr. Rebecca Auer, a surgeon scientist whose leadership and vision have made her one of Canada's most compelling voices for integrating research and care. In her widely read Globe and Mail article, Dr. Auer called on Canada to make research an integral part of care, arguing that every patient deserves access to the latest knowledge and every clinical encounter is an opportunity to learn. This vision, that research is care and care is research, has become a rallying point for change across our cancer system. Today, Dr. Auer will challenge us to think differently about how we deliver care, engage patients, and structure our institutions so that research and care are not parallel tracks, but one unified system that continuously learns and improves. Please join me in welcoming Dr. Rebecca Auer.
Thank you so much for the opportunity to speak with you today and to be the keynote. It truly is an honor. Conferences such as this, and I had a wonderful opportunity to attend for the first time last year, I think are exactly what we need to bring our ecosystem together: patients, researchers, clinicians, industry, government stakeholders, to tackle this challenging and complex problem. I titled my talk today, Research and the Courage to Hope. And what I really wanted to frame for you today was the why, as I see it, as a researcher, as a clinician, and as a patient caregiver. and not come up with all the solutions, because I think many of them have already been brought forward by so many of you in the audience, but also this next 2 days is really the opportunity for us to, I think, come together with some of these solutions. So when we think about clinical trials, we think about that transition of discoveries right through to clinical care and approved therapeutics. And as researchers and clinicians, we sort of think about phase 1 studies as that translation from the bench through to the bedside, first-in-man studies, trying a therapy for the first time. But for patients who are facing potentially terminal diagnosis, it really does feel like making a miracle. We know in some phase 1 studies, there are patients who have unprecedented responses. And so I think as a patient, when you're looking at a phase 1 trial, you are looking at it differently. And that's not to say that every phase 1 trial patient does amazingly well and every phase 1 trial leads to a market authorization eventually. But there is that hope in those early trials. I think as we move forward, we're really trying to unravel the miracle, if you're thinking about it from a patient's perspective. Why did it work in 1 out of 10 patients? Why does this patient respond and the other patient doesn't respond? And I would argue that when we design trials, this is probably where we need to invest most of our energy and time. A signal that it worked in 20% is not a reason to give it to 1,000 patients and hope that 200 respond. It's a reason to ask, why did those 20 respond and why did the 80 not? And what can we do with the 80 to figure out how to get them to respond? And then lastly, we normalize the miracle. And we've seen this. A perfect example is in MSI high colorectal cancer, which I'll talk about, where we now just understand that a certain subtype of cancer is going to do extremely well with a novel therapeutic. And that's happened all across the cancer ecosystem, where that just becomes the new norm. So I think it's time to reimagine this process of discovery, translation, and I'll tell 3 stories, a story of Adam, Camille, and Hayden, hopefully illustrate that point. I talk a lot about the power of yet and choosing hope. I've told this story before, but my son came home from school one day when he was 7, and he asked me, what's the most powerful word in the English language? I thought it might be love, which of course he laughed at me. But he said it's yet, because when you add yet to the end of any sentence, it makes the impossible possible. And obviously this is something they talked about at school. And I loved this story, and I used it in every patient encounter I had. If a patient had a metastatic cancer. There was no cure. They would ask me, Dr. Auer, is there a cure? And I would say, there's no cure yet. Because yet has actually been a realization, even in my own career, that some patients that we would have said 10, 15 years ago, there is no cure, we now say there is a cure, at least from what we can tell. I think hope is in and of itself a very powerful medicine and a powerful driver. Hope is different than wishing. Hope has action behind it. Hope usually is associated with a plan. And I tell this story about Christopher Reeve, Superman, who had the terrible horse accident and was paralyzed from the neck down. He absolutely believed for his entire life that he would someday walk again. And the power of his belief actually allowed him to have use of his hands over time with significant amount of therapy. He died, unfortunately, before he ever realized his dream, but he lived every day with hope, and he talked about how important that was, and he said, once you choose hope, anything is possible. I think that patients deserve to have hope. They deserve to live with hope, even if the outcome is the same and they ultimately die of their cancer. Living with hope is much better than living with despair. So I think the journey through your cancer diagnosis is just as important in many ways as the outcome. And supporting that journey is something that we all have a responsibility to do. So I believe in the power of yet, and I think that things change when we, not when we seek to achieve what we want, but when we never lose sight of why we want it. That's a quote from Simon Sinek, who writes the book Start With Why. And he talks about how really great companies and really great movements, they don't focus on what we need to do or how we need to do it. They focus on why we need to do it. He tells the great story about Martin Luther King, who gave the I have a dream speech, not the I have a plan speech. So when we think about cancer therapeutics, we can look back at where we have been. And many of the therapeutics, the surgery, radiation, and chemotherapy are still the backbone of many cancer treatments. And they have certainly been successful. But they come from a different time as well. And so as we progress through science, we see that although these remain the backbones of cancer therapy in many ways, there are alternative strategies. I put a picture up there of who's widely considered the father of chemotherapy. This guy here, his name is Cornelius, or Dusty Rhodes. And he was actually recruited to the Rockefeller Institute and Sloan Kettering Hospital from the Chemical Warfare Division of the US military, because he was an expert in poisons to kill enemies. And that is essentially what chemotherapy is and the origins of it. And I think this is really important to remember that that is where our therapeutics originally came from, this idea that we need to kill the cancer and hopefully not kill the patient in doing so. And of course, that has led to what we know to be a lot of the side effects of our cancer treatments that patients, even when they are cured, do live with. There's a picture there of a woman who had a Halsted mastectomy, and many, many Halstedian mastectomies were done before we recognized that it wasn't actually more effective than a modified radical mastectomy. But we see these side effects of chemotherapy, and again, the journey is as important as the outcome, and so we need to think about these things. And where have we been in terms of clinical trials? Well, this is kind of has been the standard for randomized clinical trials really since the 40s, I think, this algorithm where we have a new therapy. We test it in a phase 1, small number of patients, really focused on safety, not a lot of interest in efficacy at that point, but can we safely give it to patients? Phase 2, we talk about, does it seem to work, and what dose, what's the highest dose we can give before we make the patient too sick that it's not worth it? And then, of course, we get 1,000 participants or more, and we compare it to the current standard, and we try and figure out if it works. And this is really the traditional clinical trials algorithm. And so really there's been a division, again, in cancer, but in all diseases, between research and care. Because, of course, if you're going to give a poison to a patient, you need to have a very clear distinction between research and care. And this has come from really an important place in our history of why there are such sort of strict lines between what we consider standard of care and what we consider research. And many of you probably know this story, but I'll tell it anyway. The Tuskegee study was actually a partnered study with the US government, public health, and Tuskegee University in Alabama. It started in 1932, but it didn't end until 40 years later. And the US Public Health Service partnered, and they enrolled 600 impoverished African American men who really didn't have access to health care. And they tested all of them for syphilis, and almost 400 of them had latent syphilis. The reason to participate in this study for these men was that they would receive medical care and dental care, which they did. But they were not told that they had syphilis. They weren't told that they were tested and they had syphilis. Penicillin was determined to be effective for syphilis in 1947, but the participants were not treated because this was a study to look at the natural history of syphilis. When the study was ultimately stopped in 1972, 128 participants had died directly or indirectly from syphilis or its complications. 40 of these men's wives had been infected, and 19 children had been born with congenital syphilis. So you can imagine this is an absolute tragedy and failure of our ethical and regulatory frameworks. And this really led to the creation of the Belmont Report. And it heavily influenced what was called the Common Rule, which we still use today. And essentially, it's that clinical trials need to minimize risk. Those risks need to be reasonable. Patients need to be recruited equitably. We need to ensure there's informed consent. We need to monitor for safety, protect vulnerable patients, and maintain confidentiality. And I would argue these are still as important today as they were in the 1970s. But I would also argue that our current regulatory framework actually doesn't necessarily support this, particularly things like protecting vulnerable patients and recruiting participants equitably. We know that's not true. And so I think the principles here shouldn't change, but I think some of the frameworks that we have around clinical trials do need to change. So where are we now compared to where we've been in cancer therapies? Again, I know all of you are familiar with the incredible explosion in molecular biology and molecular therapeutics, and this is from a sort of 75-year journey in understanding really the cell biology and the molecular mechanisms that are responsible for cancer and what drive it. And when we think about the pace of change, it's really quite dramatic. And you know that we sequenced the first human genome for the first time in 2003, which is just over 20 years ago, and cost almost $3 billion, and it took 13 years in many, many countries to do it. And now we can sequence a human genome in 1 to 2 days, and it costs about $1,000. Another example is CRISPR-Cas9 gene editing, which was only discovered in 2012 in strep pyogenes, but now is in an FDA-approved therapeutic for sickle cell anemia. The pace of discovery is substantial. Monoclonal antibodies, which we know is a really important therapeutic in cancer and outside of cancer, we approve almost one every 2 to 3 weeks now. So it's really this pace of discovery that is bringing therapeutics down the pipeline into the market. Same with immunotherapies. We've got checkpoint blockades, which activate the immune system, have completely transformed many cancer cares. We also have cellular immunotherapies. We had first approved in 2010, again, only 15 years ago. This number, actually, I forgot to change it, but it's at 34 now. And there's, now, upwards of 10 therapies approved a year. So it's quite an incredible pace of change. This is just a little graphic of a T cell killing a cancer cell. And so this is what the future might look like. Now we may have 100 different therapeutics that are targeting specific molecular or immunotherapeutic pathways. But in the future, there'll be thousands, or maybe even hundreds of thousands. And You can see this just based on FDA approvals in oncology over time, and see how exponentially they've increased. So this is an incredible rate of progress that is continuing to increase. This is our clinical trial algorithm today. It remains completely unchanged. And I would argue that this algorithm is not going to meet the needs of the incredible explosion of biotherapeutics and molecularly targeted therapeutics that we have in the cancer space. And now, instead of having one patient that might benefit, it might be 1 in 100 patients that would potentially benefit from your therapy. And now trying to find these patients in a country as big as Canada to be able to do a clinical trial can be very challenging. And with all of the potentially regulatory burden associated with those clinical trials and the expense, often these trials just simply can't get done. So this is Adam's story, and it's about rare cancer, so specifically speaks to this issue around trials across the country. Adam is the father of 3 boys. He had been recently appointed as CEO of the Cement Association, and he was diagnosed with stage 4 bile duct cancer, cholangiocarcinoma. It's a very rare cancer, but 800 patients diagnosed per year in Canada. At the time, he was 49 years of age. He started standard treatment with chemotherapy, and his life expectancy was less than a year. Adam's also my husband. I was devastated. I really thought that he was not going to be there for more than 1 year of Christmases, Easters, holidays, summer vacations, and I would be a widow, and my children would grow up without a father. And John Bell, who many of you know, incredible leader in biotherapeutics in Canada, I walked into his office and I told him the bad news, and he said, don't forget the power of yet. You always told me that story, and there's a lot of hope in research. And we know it's no matter a question of if we will cure cancer, but which cancers will cure first. And maybe we need to focus a bit on cholangiocarcinoma. And that moment, remembering how research is changing the cancer landscape, was an incredible moment for me because it was the first time I had any hope. And that for me is probably the most powerful moment in this journey. Having hope, having somebody actually say, there's a possibility, however small, that your husband may not die of this cancer, was what kept me going. Adam's story is different from the average story. We were able to get molecular testing through Foundation Medicine, but also through a clinical study. That our oncologist had enrolled Adam in, and he had a mutation in a gene called BRCA, which is common in ovarian, breast, and even pancreas and prostate cancer, but pretty rare in cholangiocarcinoma, and made a rare disease a very ultra-rare disease. He got access to immunotherapy with durvalumab, even though it hadn't been Health Canada approved, because we were at a very research-intensive hospital. And so we were able to get it through a compassionate access before Health Canada approval had been sought. We started a targeted therapeutic that had been demonstrated to be beneficial in ovarian, prostate, breast, and pancreas cancer, but of course had never been tested in this rare cancer. And we went down to the National Institute of Health for an immunotherapy, cell therapy clinical trial, which unfortunately is no longer an option for Canadian patients since the new government. And because of this, Adam is doing well today, currently disease-free, almost 3.5 years later. And we have so much hope. But again, most of this stuff would not have been available or simply is not available as standard of care. Molecular oncology testing in the country is very fragmented. It's only covered if it informs the use of treatments that are already covered, so it cannot help patients. It's not covered to help patients access clinical trials. There isn't currently a standardization for which types of molecular testing are acceptable for different types of mutations, and we saw that in cholangiocarcinoma. And it's quite different across different provinces in terms of access. So this is a huge problem for us to access clinical trials in cancer in Canada. And then when you have a rare disease like cholangiocarcinoma, 800 patients, and let's say you have 5% of those patients have a mutation like BRCA, you're looking at about 40 patients a year that would be eligible even if you were able to run a clinical trial across the country. So thousands of participants becomes absolutely impossible in that type of paradigm. And so where does this leave our cancer patients? They might be lucky enough to get a site-specific clinical trial if it's a common cancer, maybe like breast or sometimes colorectal, for a molecular trial. Or there might be a basket or site-agnostic trial, so anyone with the mutation, regardless of where it comes from. Sometimes companies are willing to give compassionate access if a drug has been proven in one cancer but not in a different type of cancer. Some patients, if there's a Health Canada approved indication, can pay for that drug. We did pay for the PARP inhibitor, and it was about $9,000 a month. So not accessible to most Canadians. Most patients get no access at all, and this is where there's a real tragedy. This is Camille's story. Camille is a single mother. She'd accepted an offer to start her dream job when she was diagnosed in January 2020 with acute lymphoblastic leukemia. At the time, she was 35 years old. She was treated at Princess Margaret, received all the standard chemotherapy, stem cell transplants, all the treatments that were available. But unfortunately, the cancer recurred and there was no standard of care treatments left in Canada. At the same time, CAR T-cell therapy was completely transforming leukemia care across the United States and the world. And I'm sure many of you know, but CAR T-cell therapy, you collect the T-cells from the patients, you arm them to attack the cancer with what's called a CAR, and then you grow them up and infuse them back in the patient. And this very famous young patient, Emily Whitehead, was the first pediatric patient who was treated and was cured. And this is Carl June, who's one of the people who developed the CAR T platform. So the first clinical trials of CAR T were in 2012. By 2018, there had been 2 phase 2 studies that demonstrated a more than 50% survival in what was otherwise a 100% fatal disease. So really transformational, not a phase 3 required in this particular case. And so it was Health Canada approved in 2018. At that time, there had been 2 trials in Canada of CAR T-cell, both at Princess Margaret, with less than, I think, 5 patients enrolled all across Canada. So we had no real access to CAR T-cell therapy anywhere else. We had no clinicians who'd had experience, and we'd had no patients who'd had the benefit of CAR T-cell therapy up until that time. CAR T was also, at that time, about $1 million a patient because patients had to travel down to the US to get it. And then, of course, shortly thereafter, COVID happened, and that limited that access as well. So we had no manufacturing infrastructure and no real innovations in this space in Canada. Thanks to the work of many of these people, including the lead PI, Dr. Natasha Kekri, Stephanie, who of course is one of the co-chairs of the conference at BioCanRx, John Bell, who was one of the visionaries, and all of these other individuals, they sort of sat together and said, why can't we just make CAR T cells here in Canada? And that's what they ended up doing, created this pipeline for making CAR T cells that allows Canadians to develop the infrastructure and the expertise, provides a pipeline for our own discoveries to be able to get through, so the CAR T 2.0 can be evaluated right here in Canada and allows patients to have access and clinicians to get experience. Camille was able to access the clinical trial of the Made in Canada CAR T cell therapy. It was funded through grants and philanthropy. This was not a standard of care. She was not eligible to receive the standard of care because she was too old. The standard of care was only for patients, I think, under the age of 25, I think it is. And she is now cured, we think. She's 4 years disease-free, able to raise her daughter, watched her graduate from high school. So really an incredible story of how when we band together in Canada to create therapeutics for our own patients, we can really support the patients, but also the entire ecosystem. The last story I'll tell you is Hayden. Hayden is probably quite well known to many of you because he worked at Colorectal Cancer Canada for a while. An incredible young man and a patient of mine. He was a semi-professional soccer player. He finished his master's and was actually training with Real Madrid. He was diagnosed with a locally advanced rectal cancer in the spring of 2021. He was 22 years old at the time. He'd had some rectal bleeding. He told his coach. His coach said, "Don't worry about it." He told his mom. His mom said, "Worry about it." He was supposed to start neoadjuvant radiation and chemotherapy, followed by surgery. We know that this is pretty challenging for a young man. His tumor was in a location that he'd probably have a permanent colostomy. The likelihood of impotence and infertility was very high. And also neuropathy in his hands and his feet. He would not play soccer again. But because we had access through experimental molecular testing, we were able to test his cancer, and he had a mutation in a gene that made it what's called MSI high. And that means that he had a lot of mutations in his tumor. He had Lynch syndrome, so he had inherited this, we now know, from his mother. We know that immunotherapy works much better in cancers that have a high mutational load. And you can see here colorectal cancer with a mutation, MSI high mutation or mutation in DNA repair, responds really well to immunotherapy. And this is just a circus plot showing how many mutations there are in MSI high cancers. Each one of these red dots is a somatic mutation in the tumor compared to a non-MSI high colorectal cancer. And immunotherapy, in this case pembrolizumab, was actually approved based on phase 2 studies of 41 patients. And this was tissue agnostic, or a tumor agnostic approval, which means that this was at the FDA level. This wasn't true at Health Canada, unfortunately, but this is, I think, where we need to be thinking about in terms of clinical trial design. You don't need a phase 3 trial if you understand how a drug works, and then you only put patients in the study that you believe it should be effective for, then you can have these types of results. So these patients in blue had MSI high cancers, and you can see by this waterfall plot, their tumors were the ones that shrank. This is actually a patient of mine on a clinical trial with a drug called dostarlimab, which is also an anti-PD-L1 antibody. And this is after 6 weeks of treatment on immunotherapy. This tumor has become this little spot. So Hayden started experimental immunotherapy. It was not available as standard of care. It's still not available for someone in this case as standard of care, but we were able to access through an experimental protocol. And he started in May 2021. He had surgery in September. We now actually think surgery may not even be necessary for these patients. And he had no signs of tumor. I asked him to send me a picture of him on immunotherapy treatment to just show the contrast between what he might have looked like if he'd had chemotherapy and radiation. And like any 20-year-old, he sent me a picture of him cliff jumping in his bathing suit. But you can see he did very well on treatment. He's now just finished law school, about to take the bar, I think, if not just taken it, and is quite committed to advocacy in this space as well. In April 2025, updated results of the dostarlimab study were published, and here there was some 41 patients, and they all had a complete clinical response. Now, some of them ultimately did require surgery, but if you have a study of this many patients and you have a complete clinical response in all of them, and all of them remain disease-free, it's hard to imagine why you need more data, but unfortunately that's where we're at. And I bring this up because even yesterday we had a 40-year-old patient we're discussing at our rounds, the exact same situation. And every single oncologist in the room said if it were me or my loved one, I would take immunotherapy. But it's not funded. It's not Health Canada approved for that indication. And there's no real way to get it right now. So I do think we have to do much better here. With every single oncologist knows what they want to do, and they cannot do it. I think we need to blur this line between research and care. I think we have to maintain the principles that came out of the common rule But some of our regulations, some of our frameworks actually don't support those principles. They actually make it harder for patients to participate in clinical trials and have equitable access. So if we can blur that line and we can philosophically understand how research is such an important part of care, and no story is better told than in cancer. I think we need advocacy, a unified voice, and a single plan. It may not be a perfect plan for every single stakeholder, every single group, every single patient, but I think if we can't come together as a community and have one set of priorities that are actionable, one plan that we can actually push forward in this country, then we'll lose this amazing opportunity that we have now. This is just some pictures from our Cancer Clinical Trials Workshop in May 2024, where we brought stakeholders around the table. Thanks to Canadian Cancer Society and Stuart, who talking later today. We had our co-chairs, or not co-chair, the co-chairs of the Canadian Bipartisan Cancer Caucus as well. And I think if we can bring some of those priorities and action plans together as a group and put them forward, again, with as many stakeholders behind the plan as possible, then I think we have a chance to really make a difference. And we need to. Our patients have courage, and they deserve the hope that comes from access to clinical trials and clinical research. Thanks very much.
Thanks so much, Rebecca. My mic is on. Yes, I'm all good to go. Thank you so much. And we're all going to have a chance to interact more with Rebecca as she's leading the next panel. I'm going to ask Michelle, Jennifer, and Andreas to come on up next. And I'll introduce that panel in a moment. But before we do, I'd like to go to our next question. It's very clear, based on Rebecca's keynote this morning, why we chose this topic. There is the question we have here is, let me stand over here. What does hope through research mean to you? Turning today's discoveries into tomorrow's treatments, giving patients and families reason to believe in better outcomes, advancing knowledge that improves quality of life and survival, building a system where every patient can benefit from research, inspiring collaboration and innovation across science and care, ensuring equity so that progress reaches every community. Has everyone pretty much voted? Okay, so about half, building a system where every patient can benefit from research, which I think, Rebecca, you're going to be quite thrilled about that result, yes?
Oh,